Multimorbidity and Evidence Generation by Carlos et al. Most people with a chronic disease actually have more than one, a condition known as multimorbidity. Despite this, the evidence base to prevent adverse disease outcomes has taken a disease-specific approach. Drawing on a conference, Improving Guidelines for Multimorbid Patients, the goal of this paper is to identify challenges to the generation of evidence to support the care of people with multimorbidity and to make recommendations for improvement. We identified three broad categories of challenges. First, challenges to defining and measuring multimorbidity. Second, challenges related to the effects of multimorbidity on study design, implementation, and analysis. And third, challenges inherent in studying heterogeneity of treatment effects in patients with differing comorbid conditions. Introduction. Most older people afflicted by one chronic disease experience more than one. Considering just 5 major chronic diseases among older adults in the United States who have one, in fact, 53 to 85% have at least 2 of those 5 conditions, with the prevalence depending on the index chronic disease chosen. Doctors and other clinicians caring for these patients need to address the patient's problems in their full complexity. This includes taking into account the seriousness and severity of each patient's conditions in the context of patient goals, attending to the burdens and risk of polypharmacy and medical testing, managing functional or cognitive disabilities that might impact a patient's ability to adhere to therapy, accounting for gastrointestinal, hepatic, or kidney disease that impact the pharmacokinetics or risk of therapy or any other way chronic conditions interact, either directly or through their treatments. Yet, clinical research methods are not generally designed to study the ways conditions interact. For example, a review of trials reported in the New England Journal of Medicine found only 27% made it clear whether a subgroup analysis was pre-specified or post hoc. It should not be surprising, then, that the resultant evidence-based clinical practice guidelines may be absurdly unachievable or potentially harmful in patients with multiple chronic conditions. The large and growing footprint of multimorbidity demands an empirical evidence base that can directly inform practice through clinical practice guidelines that take conditions other than the index condition into account. The goal of this paper is to identify challenges to the generation of such evidence to support the care of people with multimorbidity and to advance a set of recommendations for consideration by investigators and others, reviewers, editors, funding agencies, policymaking organizations, involved in the creation of evidence for this common type of person. Here we focus on improving the generation, analysis, and reporting of primary data, while companion papers discuss evidence synthesis and integration and the development of clinical practice guideline recommendations themselves. Methods. To provide a comprehensive list of challenges, a subset of investigators, CW, RV, and DK, created an inventory of challenges raised by the presence of multimorbidity during the generation of evidence used by clinical practice guidelines based on a review of investigator article libraries. The scope and terminology of the list was amended at an in-person research retreat by the 9-member Multimorbidity and CPG study group. Next, we performed a structured, limited literature review that examined papers from 2 symposia on multimorbidity and all articles citing papers from the symposia identified using ISI Web of Science. Additional articles, including more recent literature, were included on an ad hoc basis. Our goal required that we survey the several topics described below, making a single systematic review unfeasible. This inventory of challenges was reviewed and critiqued at a second in-person research retreat by all Improving Guidelines for Multimorbid Patients Study Group investigators, CB, SD, DK, BL, JS, TT, KU, RV, CW. After further editing, the list was sent to outside experts, AV, KB-R, MP, for review and critique. The revised inventory was discussed at a conference on Improving Guidelines for Multimorbid Patients in Baltimore, Maryland, October 2010, attended by researchers from various disciplines, medicine, public health, biostatistics, and stakeholders from government, other payers, and industry. A goal of this conference was to encourage discussions that span the procedures inherent to evidence generation, evidence synthesis, and guideline development. The final set of challenges and recommendations was created after incorporation of ideas discussed at the conference. Results, challenges. Three broad categories of challenges were identified. First, defining and measuring multimorbidity during evidence generation for clinical practice. Second, the effects of multimorbidity on study design and implementation in both randomized control trials and observation studies. And third, reliably describing heterogeneity of treatment effect related to multimorbidity. These are summarized in Table 1 and discussed immediately below. Defining and measuring multimorbidity during evidence generation for clinical practice. Perhaps the most basic challenge in multimorbidity research is achieving clarity regarding the concept or concepts of multimorbidity that should be measured to address a particular question. Determining who has zero or just one chronic disease versus more than one depends on how the set of qualified chronic diseases are selected. There is currently no standard list of diseases to be considered in a disease count measure of multimorbidity. In addition, non-disease specific conditions or measures of function, hearing, vision, gait, continence, etc., are likely to be relevant for a given research question and may be desired as part of a multimorbidity measure or a complexity measure. Thus, substantial scientific judgment, which is to say subjectivity, is necessarily used to measure multimorbidity. There are numerous multimorbidity measures that have been proposed to capture underlying sickness. As a group, they present a bazaar of indicators of disease severity, non-disease specific conditions, functional status, receipt of treatment and utilization, and other items. It follows that the criteria used to validate these pre-tested measures are also varied. They include prediction of mortality, healthcare utilization, institutionalization, functional status, quality of life, and more. Thus, the selection of an appropriate measure should be driven by an explicit rationale for whether multimorbidity is being measured to study specific hypotheses, identify treatment effect modification, adjust for confounding, improve prediction, or for another purpose. We are unaware of any general framework that would help reduce the analytic complexity of the potential interactions between an index condition and comorbidities. In the case of diabetes, a framework has been proposed that categorizes comorbidities into typologies, including clinically dominant conditions, concordant versus discordant conditions, and symptomatic versus asymptomatic conditions. While such typologies may ultimately prove useful for prioritizing care, it is at this point difficult to see how it might be applied to help reduce the analytic complexity of interaction effects potentially at play in comparative effectiveness research. Multimorbidity-related effects on randomized, controlled trial, or observational study design and implementation. While the trial is the most reliable method for obtaining a causal estimate, practical and ethical considerations may preclude the conduct of trials in certain types of people with multimorbidity, particularly after a narrowly focused trial has established the efficacy of a treatment. Therefore, it is essential to consider observational studies as a source of evidence for people with multimorbidity. Observational studies are essential given the practical limitations of trials, but the absence of a focused design leaves them highly susceptible to multimorbidity-related sources of systemic bias, including measurement error and missing information on treatments, outcomes, and prognostic variables. Further, the treatment selection process is extremely difficult to understand in observational studies, i.e., how treatment choices are made by or made for the person are only partially decipherable, and the presence of multimorbidity can exacerbate confounding by indication. While prospective cohorts have the potential to address some of the concerns regarding measurement error and missing information that often mar other types of observational studies, it does not fully address treatment selection bias. Multimorbidity can affect the internal and external validity of evidence generated by trials and observational studies. It can affect internal validity by increasing misclassification of treatment or inaccurate measure of treatment intensity, by influencing selection of treatment, leading to confounding by indication, by contributing to misdiagnosis of outcomes, by interactions due to concomitant treatments, by modifying treatment responsiveness and/or treatment effects, or by increasing the likelihood of loss to follow-up. In addition, multimorbidity provides challenges to external validity by affecting selection of participants from the at-risk population, by association with adherence to treatment, and by altering the real-world effectiveness of treatments due to harms and competing risk. Heterogeneity of treatment effect. The fact that people with multimorbidity have multiple chronic conditions, each with a spectrum of subtypes and severities, as well as different treatments, means that people with multimorbidity have more ways to be dissimilar both to one another and to people without multimorbidity. When these differences meaningfully affect the risk of harm from a treatment and treatment benefits, it can be difficult to sort out this balance for an individual. For example, given just 2 chronic diseases and a single treatment for each, there are 16 possible exposure patterns, half of which may be relevant for clinical practice guidelines, and 4 relevant interactions: disease A versus disease B, disease A versus treatment B, treatment A versus disease B, treatment A versus treatment B. Examples of interactions in each of these categories with the potential for clinically meaningful consequences are shown in Table 2 for chronic obstructive pulmonary disease. So let's have a look at Table 2 on the right here. So between disease A and disease B, the example provided is the presence of congestive heart failure may exacerbate shortness of breath, altering measured effect of therapy on COPD. Between disease A and treatment B, the example provided states that COPD may be worsened by a beta blocker medication for congestive heart failure or hypertension, altering the measured effect of therapy. For the treatment A against disease B, the provided example states that steroids for COPD may worsen diabetic hyperglycemia or osteoporosis, altering the side effect profile. And for treatment A and treatment B, the example provided states that the index therapy affected by another therapy, theophylline levels for COPD may be increased if co-administered with cimetidine, altering efficacy and safety. To account for all possible disease and treatment interactions in a real clinical population is analytically impractical if the goal is hypothesis testing rather than exploration. A comprehensive list for just a single condition would require a major effort, though the task might be somewhat simplified by studying the most prevalent chronic condition patterns in index conditions. Given the myriad ways that co-occurring chronic conditions might influence treatment effect, it should not be surprising that complex patients with multimorbidity are often excluded from clinical trials. They may also be excluded from trials through recruitment failure or by cautious investigators. For efficacy trials concerned with limiting toxicity for relatively untested therapies and with measuring treatment effect under conditions suited to optimal assay sensitivity, inclusion of multimorbid patients is often scientifically justified. However, it cannot be assumed that treatment effects for highly selective efficacy trials apply to people with multimorbidity. Indeed, if such an assumption were valid, there would be little motivation or justification for excluding such people in the first place. There have recently been increasing calls for more pragmatic trials that enroll people more representative of those treated in clinical practice. It is widely believed that the results will be more generalizable to real-world routine clinical practice. However, the results of such trials have to be interpreted carefully. Recent work has underscored that in the presence of clinically important heterogeneity of treatment effect, the summary results of a clinical trial can be misleading regarding many included patients. Including people with multimorbidity is likely to exacerbate the problem of within-trial heterogeneity of treatment effect for the reasons discussed above. Indeed, the argument for including such people in clinical trials is precisely that the effect size for benefit or harm measured in healthier people may not apply to those with chronic diseases. Including subpopulations within a clinical trial where substantial heterogeneity of treatment effect is anticipated may thus yield a single summary result not as directly applicable to any individual as in a homogeneous trial sample. Table 3 shows that the sample size required to test a treatment effect interaction hypothesis increases by a factor of 4 to 5 if half the sample has a chronic disease of interest, and by a factor greater than 10 if only 1/5 does. Testing more than one multimorbidity-related subgroup effect requires still larger samples. Heterogeneity of treatment effect. Since people can have so many different combinations of multimorbidity, and because any additional chronic condition or multimorbidity pattern can potentially influence the risk and benefits of treatment in different ways, the conundrum of multidimensionality is especially relevant when considering the evidence base for people with multimorbidity. While recommendations and frameworks for analyzing heterogeneity of treatment effects have been proposed to partially address these issues, as yet there is no clear consensus on how best to cope with heterogeneity of treatment effect in the context of clinical trials, and in many, perhaps most circumstances, it will simply not be possible to address every potential clinically important multimorbidity-related question in the context of a randomized trial. So this table on the right shows the sample size required to achieve adequate power for main effect versus interaction effect. And so we see if the proportion of sample with multimorbidity is 0.2, so if 20% of the sample has multimorbidity and the number of subgroup analysis is 1, then the sample size needed for test of main treatment effect is 800. The sample size needed for test of treatment effect modification by multimorbidity is 9,700. And the sample size multiplier to study multimorbidity interaction B over A is going to be 12.1. Go ahead and take a moment and go through the remainder of the table. Recommendations. Based in part on the barriers identified above, the Conference on Improving Guidelines for Multimorbid Patients provided a dedicated opportunity to discuss the recommendations for how evidence generation might be modified in order to best inform the downstream processes of evidence synthesis and guideline development. Our initial set of recommendations focused on actions researchers should currently consider when performing a trial or observational study to make their results more relevant to the multimorbid patient. These directly address the challenges identified above. However, we recognized that adhering to these recommendations was likely to have only a relatively modest impact on the evidence base. We therefore proposed research priorities to address critical methodological issues and a map that points to infrastructural changes necessary for more robust evidence generation. Even though these recommendations were derived from a conference of experts from a variety of fields, they are limited by the need to cover a number of relevant topics. Had it been possible to conduct formal, systematic literature reviews for all of these topics, additional challenges and recommendations may have been identified.